Welcome everyone. Welcome to the lecture on the topic of transcription in prokaryotes. So today we'll be discussing in depth about the transcription process, the process of synthesizing RNA molecule from a given DNA template. Now transcription refers to the formation of RNA molecule using a template DNA. So what we'll be learning in this particular topic? See, we will be in the entire lecture, we will be focusing primarily on understanding the path of formation of RNA molecule. What are the players involved, how the enzymes are working about this particular reaction, how the enzymes are involved in recognizing from where we have to start the transcription process, and after recognizing how they are initiating about. So we'll be able to, we should be able to understand the complete process of transcription in prokaryotes only, and also we should be able to establish the understanding of the different proteins that are involved in this particular scenario. So I expect that with this lecture you will be able to learn the mechanism as well as the functions of the proteins that are involved in this particular execution of synthesis of RNA molecule. So let us begin this lecture that what is transcription. See, transcription, as I told you in brief, that it's a formation of RNA molecule. It's a formation of RNA molecule from a DNA strand. So if we talk about the genome is containing your DNA. Now the genome is very large. The genome is very large. It is getting replicated. The question is whether the complete genome is leading to the formation of RNA molecule. The answer is no. Only a small stretch of DNA codes for RNA molecule. The genes which are actually required for the formation of proteins, and the pseudogenes which are not leading to the formation of protein but are responsible for the synthesis of RNA molecules, right? Ribosomal RNA, like tRNA, like miRNAs. So all those genes which are leading to the formation of RNA molecules are clubbed in a single category called as structural genes. These are called as structural genes. Whereas the genes which do not code for any mRNA are clubbed in the category of regulatory genes, and those genes are responsible for regulating this expression. So broadly we are dividing into two categories. One is structural genes which will eventually lead to the formation of mRNA. Others are regulatory gene which will regulate this process of transcription. And why transcription is so relevant? Because in every kind of response, whether it is normal physiological conditions or it's a stress, it's a response against any kind of stimuli, any kind of stress, any kind of change, be it environmental, be it any physiological change, the first thing the body is responding by synthesis or varying the protein levels, and then that variation is mediated by the transcriptomic level, and that transcriptome or transcription gives rise to mRNA. That's why it is very important for us to study. That's why it is very important for us to study. So keeping that in mind, the main question that comes first is, we have double-stranded DNA. We have a double-strand DNA. One is in 5' to 3' direction, and then another one is 3' to 5' direction. The question is, out of these two, the double-stranded DNA, out of these two strands, which strand will be used for the synthesis of RNA? Whether it is single strand or whether it is both the strands, or if it's single strand, which orientation shall be used? So based on that, let us focus on this particular image. See, the image is clinical representation of double-stranded DNA, where the leading strand, or I should say 5' to 3' is in black, 3' to 5' is in red. Now transcription is happening in this stretch, which is giving rise to a stretch of RNA molecule, which is represented in 5' to 3' direction, and the sequence is highlighted in blue. Now let us understand this. See, let us first match the sequence of RNA with these DNA strands. Let us match blue with black and blue with red. So what we'll be getting? We came to know after matching, see, GCAGU, GCAGT is just a difference of T and U. That's the basic difference between DNA and RNA. Rest of the sequence is same. So we know that by matching that the sequence of newly formed RNA molecule is exactly identical, I should say, or similar to the upper strand, that is 5 prime to 3 prime strand, and that's why this strand is called as coding strand. This strand is called as coding strand, or it can also be called as sense strand, which will actually make a sense protein or a sense mRNA. What about the other strand? If you look carefully, the other strand is complementary to that particular RNA synthesis. That means in order to replicate the coding sequence in form of RNA, the template that should be used is nothing but a complementary strand, and that's why the 3 prime to 5 prime direction DNA strand is used for the synthesis of RNA molecule, and that's why we are calling it as template strand. And this template strand is actually called as antisense strand. Why? Because it is complementary to the actual strand of RNA, actual sequence of RNA. So you see what is happening. There are two strands of DNA. Out of those two, both are not being used. Only single one is used. And which one? The strand which is actually complementary to the coding sequence, which is actually complementary to the coding sequence. So please keep in mind, students, please keep in mind, whenever a question is coming on your way in this scenario, you just have to remember one thing, that in a double strand DNA, the sequence which is moving from 3 prime to 5 prime direction, that strand will be used as template strand, and that is the strand which we are calling it as antisense strand for the production of RNA molecule. So that is clear. Now I told you another thing, that out of these two strands, only template strand 3 prime to 5 prime is being used for the synthesis of RNA molecule. That's why, that is the reason why we call transcription as asymmetric transcription. Why? Because it is using only a one strand, whereas the opposite strand is not being used. That's why we are naming it as asymmetric transcription. Now consider a situation where there is a double strand DNA and we are talking about the transcription process, and the transcription process is happening at two places. The transcription process is happening at two places. Fine. The first one, the left hand side, is moving from 5 prime to 3 prime direction, left to right in your panel. The other is moving right to left. Please remember, the synthesis of mRNA is always happening in a 5 prime to 3 prime direction. No matter whether it is moving in left to right, no matter whether it is moving from right to left. I told you a bottom line or a key factor, that a strand which is 3 prime to 5 prime as per the direction of movement will always act as template strand. Now if I consider a transcription bubble moving from left to right, then the bottom strand which is 3 prime to 5 prime, it is acting as template. If the bubble is moving from right to left, this according to this black arrow, right to left, then this direction is 5 prime to 3 prime and the other strand direction is 3 prime to 5 prime. Then this strand will be used as template strand. So in prokaryotes, when we are talking about the movement of the transcription, always remember the direction wise, that the direction wise 3 prime to 5 prime direction is of utmost important, and the DNA strand which is 3 prime to 5 prime direction as per the movement of replication curve will always act as template strand, will always act as template strand. So let us, after establishing that what strand is being used for the transcription, let us look at the major player which will cause the process of RNA polymerization, and that major player is nothing but an enzyme. Not even enzyme, it's an complex, it's an enzyme complex called as RNA polymerase. RNA polymerase. The prokaryotes are having only a single RNA polymerase. Please remember, it contains only a single RNA polymerase. But that RNA polymerase is a complex enzyme. It is made up of 4 types of proteins. It is made up of 4 types of units. Now, please listen carefully. I have told you 4 types. How many number of units? If I am talking about in terms of number, then there are total 5 units. How? See, there is alpha, beta, beta prime, and sigma. These are called as subunits of this enzyme complex, that is RNA polymerase. There are 2 units of alpha, 1 unit of beta, 1 unit of beta prime, and 1 unit of sigma. Cumulatively, there are 5 units. But when you're talking about the type, then there are 4 types: alpha, beta, beta prime, and sigma. So please read the question carefully. When we are talking about, when somebody is asking you for the type, that means there are 4 types of subunits. When I am talking about, when I am talking about the number of subunits, then there are 4 number of subunits, because alpha is present as a dimeric form. There are 2 subunits of alpha. Now what are the functions of these subunits? How these subunits are held together to give rise to RNA polymerase. We know that RNA polymerase has just one activity, that is polymerization of RNA. Polymerization of the monomeric unit, that is called as ribonucleotide, forming a phosphodiester bond between the 3' OH and the 5' phosphate group of the incoming nucleotide. That is the major function of this RNA polymerase. That's why we call this RNA polymerase as DNA dependent RNA polymerase. DNA dependent RNA polymerase. So what are the roles of this? What are the functions of these subunits? Let us see. See, beta prime. Beta prime is responsible for binding to the DNA. It is responsible for the binding to the DNA. The beta subunit. The beta subunit is the main polymerase unit, is the main catalytic site for polymerase. This beta unit is the main polymerase site. The sigma factor, it is one of the most crucial part of this particular holoenzyme that I am talking about. The sigma factor is responsible for initiating the transcription. It will recognize the promoter sequence. What are promoter sequence? There are certain sequences that we'll see in the forthcoming slides. There are certain sequences which are present upstream to the start site, upstream to the start codon, and those sequences are responsible for promoting the transcription. That's why the term promoter was coined to them. So recognizing where the promoter is and preparing for the transcription, signaling that yes, right now you have to stop and you have to look forward towards the transcription. That's the job of your sigma subunit. Then there is alpha. Alpha is responsible for holding everyone together to form an enzyme complex. All the subunits, the integrity of this particular complex is maintained by alpha subunit. Not only the integrity, but it also determines, the alpha subunit also determines the template which needs to be used for the synthesis of RNA. So you see, the alpha is responsible for holding the assembly as well as recognizing the template DNA. The beta is responsible for polymerization. The beta prime is responsible for binding the DNA, as also it has the helicase activity which will open the double strand DNA, because you need to open the double strand in order to get an excess of a template strand. Once you get the excess of the template strand, then and then only an RNA molecule will be synthesized using this. And sigma, as I told you, requires for the initiation of this particular stages. Then based on this preamble what we have studied, let us now start discussing the stages of transcription. See, it's very common. The stages can be what? Once the transcription has to be initiated, then it should be elongated, then a termination should happen, as we had discussed there in the case of replication process. But I have added a pre-stage also. What is that pre-stage? Let us see this. See, we should also be able to understand that how that RNA polymerase complex is actually binding to the promoter site, how it reaches the promoter site, whether it directly comes from the cytoplasm and attaches on the promoter site, or there is a certain kind of scanning mechanism in order to find this particular promoter site, and then what happens next. So based on that, I have divided this particular transcription into 4 different stages. First is your binding of RNA polymerase. Second is your initiation of this particular transcription. Then there is chain elongation. Then there is chain termination. So let us focus on the first part. That is binding of this particular RNA polymerase to the sequence. See, this pictorial representation is a cumulative representation of the complete transcription process. Let us come to this at the later stages, and let us focus on a main part that I want to tell you. See, once there is a binding, then this particular strand is getting opened. Now it's a double helical structure. Please remember, this is DNA is a double helical structure. So if I am opening a short segment in between, then there will be supercoiling which is induced here as well as in the front or in the downstream. So it needs to be relieved. the supercoiling needs to be relaxed, and that is the function of topoisomerase. So while the transcription is going on, the topoisomerase also comes and performs its function, thereby relieving the torsional stress because of the opening of double-strand DNA. And in this opening in this transcription bubble itself, the RNA will start synthesizing. The new NTPs, the new ribonucleotides will be coming, and the old chain will be coming out of the bubble. The newly formed chain will be attached as RNA DNA hybrid, and that is our job to study. Let us focus on the first stage, and let us go step by step. See, binding of the RNA polymerase. Binding of RNA polymerase to what? To the template DNA. To the template DNA. How it binds? See, polymerase has no specificity first of all. So it will never directly go and bind to the promoter. Because if it is going directly and binding to the promoter, then we should have said that is specific to the promoter. No. It can bind to any region of the DNA. It can bind to any region of the DNA. And the binding is of very, very low affinity. It will bind the DNA and it will start migrating on the DNA. It will start scanning the DNA where the promoter is present. If the promoter is present here, it will start scanning the DNA. It will reach to the promoter and then it will halt. And then it will halt. Why it will halt? Because the binding of DNA is mediated by alpha subunit. It started scanning. The sigma unit is also present. The sigma subunit recognizes now that this is the promoter. Please stop. We have to prepare for transcription. While the RNA polymerase is binding, while the RNA polymerase is bound to a DNA molecule, RNA polymerase is bound to a DNA molecule. It's a closed complex. Once it reaches to the promoter and initiates the transcription, at that time it is opening the double-strand DNA, and that complex is called as open complex, open promoter complex. And at that time, the binding stability is very high. The dissociation coefficient is very low. So what are the promoters? What are the properties of the promoters? How many type of promoters are actually present inside the prokaryotes that are leading to this particular transcription initiation? Let us look at this particular region. See here in this picture. The plus, this is a double-strand DNA. The plus one is the first nucleotide that will be added on mRNA, the first site. This is called as start site. And the transcription is processing in this direction. So the bottom strand is the template. DNA. Now, when we are saying the word upstream means all the segments of the DNA which are ahead of this particular start site to the left hand side. That's why we have termed as minus. Minus means upstream. So there are two promoter sequences. Promoters are nothing but a very short stretch of DNA sequence. There are two promoter sequences which are present at minus 10. Minus 10 means 10 base pairs upstream to the start site, and at minus 35 base pairs ahead of the start site. Now the first promoter that is present, that is minus 35, is called as GACA box, that is TTGACA. This sequence of promoter is conserved throughout prokaryotes. Right now we have taken an E. coli as a model, but it is conserved in all kinds of prokaryotes. Then this particular promoter sequence, after this there is another promoter sequence called as Pribnov box, which is unique to the prokaryotes, and it is also conserved throughout prokaryotes. Now the sequence is TATA, TATA-AT, and the analogous to this is also present in the eukaryote, which is called as TATA box. In prokaryotes, it is being called as Pribnov box. Fine. So these are the promoter sequences, as you have seen here. These are nothing but the short stretches of DNA which will actually function in promoting the transcription of this particular DNA sequence. Now, suppose somewhere here the RNA polymerase has bound. It will start scanning the double-strand DNA. It reaches to the minus 35 position, and then it realizes, oh, the sigma realizes that this is the promoter sequence. Now we have to start synthesis of mRNA molecule, and that's the job of the promoter. Fine. Then let us go to the next stage, that is initiation of polymerization. How that initiation of polymerization is happening. See, initiation of polymerization is nothing but you have to start with the first nucleotide. The first ribonucleotide is what you have to start with. That is called as initiation site. The +1 sign which I have told you in the last slide. Now, the RNA polymerase does not require any primer. Like in replication, what we have studied, that there is a need of a primer. That's why primase was there. Here, there is no need. The RNA polymerase is able to synthesize RNA molecule from scratch. So the first base that it adds is, preferably, the RNA polymerase prefers to add purines. It prefers to add purines, whether it is ATP or GTP, and most of the time it is GTP. Most of the time it is GTP. Once the first GTP molecule, the guanine nucleotide, is being added, then its 3' OH will be used for forming the phosphodiester bond, and the chain will continue, and the chain will continue. This is how the complete initiation process is starting. Now, what is happening in the initiation process? Once the RNA polymerase has come to -35 position GACA GACA box, that is called as promoter. Once it has come there, then it will prepare RNA polymerase. Please get ready. We are about to start the transcription, and then it will slide ahead, and then it will reach to Tata box. That is Primnov box, TAAT. The sequence I told you present at -10 position. Once it reaches to the -10 position, it starts unwounding the DNA, starts opening the DNA. So the opening is starting from the first promoter, that is -10. Who is opening this particular double strand DNA? The beta prime unit, which is having an helicase activity. Now it opens up to form a short stretch of a bubble kind of appearance, to form a short stretch. This region which is opened now is of the length of 17 nucleotides, 17 base pairs. So you see there is a short stretch where this double strand DNA molecule has turned into a single strand DNA molecule, and that stretch is of 17 base pairs. Now when I'm talking about 17, suppose at the starting there was -10, so we are 10, and then we have entered in 7 of the coding nucleotides. So see, now this initiation has started. Now the initiation has started. It has given the template an opportunity. It has opened up the template. It has given the polymerase an opportunity to add the first nucleotide at first position. Fine. And then polymerase recognizes that, and it brings the purine triphosphate, that is GTP preferably, to the site, and it adds GTP on the +1 site. Once the GTP is added, so at that particular moment, what is the complex? There is an RNA polymerase, there is a template DNA, and now a nucleotide, RNA nucleotide, has come and has been added on the first site. And at that particular time, the complex of these 3 molecules is called as transcription initiation complex. It is called as transcription initiation complex. Now, this is the end of your transcription initiation. The only thing that is remained now is to add the nucleotides and move ahead. The bubble will start moving ahead. The first opening is of 17 nucleotides. Now, out of those 17, the 10 has gone in the promoter part, and at the +1 we have added a GTP. Now the remaining is 6 nucleotides. It will start synthesizing those nucleotides as per the complement. Once a short stretch of that nucleotide is finished, then the transcription bubble will move ahead to perform the elongation part. But before moving ahead, what it does, it removes the sigma subunit now, because sigma subunit is not required anymore. What was the function of sigma subunit? It was required only for the initiation part, because it was able to recognize the promoter sequence. Now it should be released from that part, so that the beta subunit, the polymerase, can perform its function. So, and that's what is happening. As soon as a short stretch of RNA molecule is synthesized, the sigma subunit is released from the complex, and now the complete RNA polymerase goes into the elongation part. So this is just to summarize you that what has happened till now. What has happened till now? We have a RNA polymerase. We have a -35 promoter, we have a -10 promoter, and we have a +1. That means the first nucleotide, the starting part, the starting codon. Now, when the RNA polymerase binds to a DNA, it's a closed complex. Once it reaches to the -10, then it starts opening the DNA. It has created and opened up a DNA. This is called as open promoter complex. Once the open complex is formed, then transcription is initiated. It will recognize the first site and it will add the guanosine preferably at this particular site depending upon the complementary. Once a short stretch is now synthesized till here, short set stretch is synthesized, the sigma subunit is now released, and then this particular RNA polymerase now will start synthesizing the rest of the RNA molecule. Now that window of 17 is moving ahead. Suppose the window has moved now to another 17. So then 17 plus 17 is 34. But the bubble is allowing only 17 nucleotides to get open. So what is happening? Once the bubble is moving ahead, the DNA fragment which was opened in the earlier formation will now close and return back to a normal condition of double strand RNA or double strand DNA, and the RNA which is formed is protruding out of the transcription bubble. Then what is being done in the elongation? In the elongation, there is no requirement of the sigma factor, so the core polymerase is devoid of sigma factor. The rate of the reaction is very high, almost around 20 to 50 bases addition per second, and it is highly accurate. Accurate in a probability of one error in 10,000. Now, you may say it's a very high probability, but when we are talking about in terms of the rate of formation of RNA molecule, because a single gene give rise to multiple transcripts, in that scenario it's very less, and that's why it is highly accurate. So in nutshell, after removing the sigma subunit, what is happening? There is a RNA strand to which ribonucleotide triphosphate will be coming. There will be a phosphodiester bond between the phosphate and the hydroxyl group, and two phosphate groups will be removed in the form of pyrophosphate. And that's how the transcription bubble keeps on synthesizing the RNA sequence. And the nascent RNA which is being formed is continuously released from the transcription bubble, and this is how the complete representation occurs. See what I meant by release of that RNA molecule. This is the RNA polymerase. It has synthesized this particular portion. Currently, it is synthesizing this particular strand. So the strand which is being synthesized earlier is now coming out of the transcription bubble, and the portion of DNA is now winded back to form a double strand DNA molecule. So you see in transcription bubble, there is a single strand DNA and there is a RNA DNA hybrid. Only in that region, only in that transcription bubble which has opened for 17 nucleotides, there is a RNA DNA hybrid. And the replication bubble is similarly moving ahead. Now it will finish up, then next 17 nucleotides will be open. They will be forming the double strand of RNA DNA hybrid, and the remaining portion will be coming out. And this is how the complete elongation process is going about. Then the last part in this particular process of forming the RNA molecule is nothing but your termination. Now you have to terminate. There are certain stop codons there to terminate, but what happens, how this termination is brought about? Again, the termination is brought about by two mechanisms. The first is Rho termination method. Now there is a specialized protein called as Rho protein. Now this Rho is nothing but it's a kind of a helicase. Enzyme which requires ATP. It requires ATP molecule to open up a double strand structure. If this protein is involved in termination, then we call the process as Rho dependent termination of transcription. Whereas the other methodology or the other mechanism is nothing but there are certain specific sequences which are formed in RNA molecule which gives rise to stem loop like structure, which eventually changes the conformation of RNA polymerase, causing the dissociation of that particular enzyme. So let us see how it is happening. Let us see how it is happening. The row dependent and independent manner. See, this particular example is of row dependent termination of transcription. What is happening? This is the replication bubble. Suppose it has reached to a stop codon. Now, what will be happening? This RNA, which is coming out of this bubble, is recognized at the terminus by this row protein. The terminal portion will be recognized by row protein. The hexameric in the row protein will bind to this RNA in an hexameric form. Six units of row protein will be binding to it. It will move from this position and will go to the transcription bubble. It will move through the RNA molecule and now it will reach to the transcription bubble. It will reach to a transcription bubble. Once it reaches the transcription bubble, remember, it is a helicase. It will open up the double strand. Where is the double strand? The double strand is between the produced RNA molecule and already available template DNA. So it will utilize the ATP, it will perform the ATPase activity, it will hydrolyze the ATP, gain the energy, and with that energy, it will open up the RNA DNA complex. Once it opens up the RNA DNA complex, what is happening? The transcription bubble integrity is being lost. Now this particular RNA molecule will be released and the RNA polymerase will be dissociated and that complementary strand of DNA will rejoin to give rise to double strand DNA. And this is how the complete process is terminated. So what is happening in row factor mediated termination? The helicase activity of that row protein is responsible for opening the RNA DNA hybrid inside the transcription bubble by utilizing the ATP molecule, and that's how the termination is being achieved. Then there is a row independent termination in which there is no row protein involved. How it happens? See, sometimes what is happening is that the RNA molecule which is being synthesized, the last segments of those RNA molecules, the stretch of 30 to 40 nucleotides contains GC repeats, contains GC. Now they have a specific sequences like GC will be repeated, then multiple uracil residues will be added on. Now what is happening to that particular group? Because of the repeats and the complementary regions that is being induced in this particular RNA molecule, the released RNA molecule will form a loop-like structure. See, it is forming a loop-like structure. Why? Because of the addition of complementary regions. And as soon as it forms a loop, it is giving rise to a short stretch of double-strand RNA. Although technically it's not a double-strand RNA, mind you, it's just a secondary structure that is called a stem loop structure, which is folded because of the complementary. And this folding results in change in the conformation of this RNA polymerase. Now, this RNA polymerase, which was stably bind to the double-stranded DNA, is now unstable and will dissociate from the DNA, thereby terminating the transcription. So, the stem loop-like structure which are being formed because of the complementarity, they are reducing the stability of RNA DNA hybrid complex. They are reducing the stability of RNA DNA hybrid. Thereby, they cause the transcription complex to dissociate. And eventually, everything comes to normal. The DNA again rejoins to form double-strand DNA, and the RNA molecule is released for further processing. Among all the base pairings which we are seeing that are unstable, majorly the U and A unpairing. The U and A unpairing is the most unstable pairing. The ribonucleotide, the ribose sugar uracil, and the deoxyribo ATP in the RNA DNA hybrid is the most unstable pairing. And this is how, students, the complete process of transcription in prokaryotes is moving ahead. We had seen that what are promoter sequences. We have seen what type of promoter sequences are there. How RNA polymerase is binding. It is scanning. It is halting at one promoter. Then it is scanning to it is sliding to another promoter. That is Prignow box. It start opening from the Prignow box. The bubble is formed. The bubble is of the stretch of 17 nucleotides, and then it starts initiating. At the first site, it adds a purine, whether it is A or G, preferably G. Once it adds, then we are calling it as transcription initiation complex. and then the remaining 4 or 5 nucleotides are synthesized. Once a short oligonucleotide is synthesized, the short RNA is synthesized, the main sigma factor is released. Now it's the job of beta unit of RNA polymerase complex to start polymerizing. It starts elongating. It continues to elongate till it reaches to the termination part, the stop codon part. Once it reaches to that particular termination part, there are two pathways, rho dependent and rho independent. Rho dependent is depending upon the rho protein. Rho protein is what? It has two types of activity. One is ATPase, another is helicase. It will bind to the to the newly formed RNA molecule. It will travel through that single strand RNA molecule and will reach a site where there is a double strand, hybrid, that is RNA and DNA, and it will utilize the ATP molecule to open this RNA DNA, remove the RNA outside integrally, and then let this particular template bind to the principal coding strand and normalize the process and terminate the process. Whereas the other mechanism is mediated by the formation of secondary structure because of the complementary induced because of the addition of multiple GC-rich regions, and that stem loop-like structure results in the change in the conformation of RNA polymerase, which results in the dissociation of RNA polymerase, thereby terminating the replication. So this is, in a nutshell, the complete scenario of prokaryotic replication. Now let us see how the MCQs are attained from this particular, the basic questions that are coming from this particular topic, like the process of formation of RNA from DNA. It's a very simple process. Then which of the following is true for the RNA? If we are talking about its polymerase activity, what is the DNA dependent DNA synthesis, whether it is involved in diet repair or what kind of component. Then what is Pribnov box? Whether we are talking in terms of its activity, which of the following transcription termination technique is used that has RNA dependent ATPase activity. Then in eukaryote, in order to initiate transcription, what should be done when we are talking about prokaryote? So these questions will prompt you, will induce the curiosity in you to provide a link between prokaryotes and eukaryotes, so that you can easily grasp the what I've told you, and you should gain a curiosity, to read a bit more, to gain something more out of your curiosity, depending upon today's lecture. And that is how these particular MCQs are here for you, given for you, just to summarize what we have read till now. And for your further reading, these are the links that you should focus on. The lectures, the research articles, the review articles, and preferably the standard books where the illustration of transcription is amazingly good. You will have a wonderful time reading all these articles, reading all this literature. So with this, students, thank you so much for your patience and patient listening, and I hope the lecture was fruitful for you. We'll meet again in another lecture. Till then, please stay safe.